Father, Son, and the Holy Spirit. God, you are the creator and the sanctifying Spirit. Help us to carry out in our lives the love and example of Jesus by striving for peace within ourselves and seeking to be peacemakers in our daily lives, by accepting suffering in the struggle for justice rather than inflicting it, by refusing to retaliate in the face of provocation and violence, by persevering in nonviolence of tongue and heart, by living conscientiously and simply so that I do not harm creation or deprive others of the means to live, by actively resisting evil and working nonviolently to abolish hatred and causes of hatred from our hearts and from the face of the earth. God, we trust your sustaining love and believe that just as you gave us the grace and desire to offer this, so too will you bestow abundant grace to fulfill it. We pray for peace in your name. Amen. Father, Son, Holy Spirit, amen. Again, just kind of a few shout outs on our program. You'll notice just one mentor, but a really important one for us, Dr. Joseph Hulbert. I'm not sure if he's here this evening, but he kind of new for us, a new tap for us, which we're really excited about. I didn't thank kind of one of the more important people. You'll notice on the instructors, I'm listed second, even though I'm the director. But the dude who started this all back, Ron, was that 36 years ago? Something like, yeah. So he's responsible for all this. So really, if you have any complaints, go to him. If you have any compliments, go to me. But I just want to give him a hand for starting this. And he's been at Bellarmine for has it been over 40 years? 48 years. So he probably only has another 30 years left here at Bellarmine. So we're so fortunate to have him around. So in any case, well, we're going to get started. And we're going to start with Maya and Lauren, and they're going to talk to us about something really important. So come on up.
Hello, my name is Maya.
I'm Lauren.
And our research is on determining the accuracy and efficacy of date rape drug detectors.
Every year, thousands of people are victims of rape, specifically drug-facilitated date rape, which occurs most commonly in college party settings and bars in the United States, but throughout the world as well. Drug-facilitated date rape occurs when a person is drugged and then sexually assaulted. Typically, the perpetrator drops a pill or powder into a person's drink to drug them. There are many substances that may be used to take advantage of the victim by lowering their awareness and making them lose control of their own bodies. Part of the reason we chose this research project is because this is a prevalent issue at most universities, and we are women entering college soon, so we wanted to educate ourselves further on this topic and share our findings with others to spread awareness. While researching this subject, we found that drug-facilitated rape is the most common violent crime at universities, and 80% of victims are women. We also found that women in college between the ages of 18 and 24 are 3 times more likely to experience sexual violence.
One of the most commonly detected drugs used for date rape is ketamine. Ketamine is a dissociative anesthetic that doctors may prescribe for depression, anxiety, status epilepticus, or sedation. Ketamine causes a sleep-like state, but comes with side effects including hallucination, agitation, and confusion. When ketamine is mixed with alcohol, the side effects can become even more extreme. Ketamine takes effect almost immediately, and there is no way to know if the drink has been spiked without experiencing the effects, because this drug is tasteless, colorless, and odorless. In recent years, commercial products have been put on the market in hopes of alleviating this issue. Detectors are advertised to, quote, detect the possible presence of illicit date rape drugs, GHB and ketamine, in alcoholic and non-alcoholic beverages, end quote. Past researchers have evaluated the accuracy of detectors based on the product's limit of detection at a specific concentration of ketamine, but no studies have been done to determine if changing the concentration of ketamine in the liquid affects the accuracy.
To bridge this gap in knowledge, the question asked in our study is how are coaster and test strip date rape drug detectors affected in accuracy and efficacy by change in concentration of ketamine and change in liquid? We hypothesized that the test strips and the coasters will have the same accuracy, that changing the concentration of ketamine will affect the accuracy of date rape drug detectors, and changing the liquid will not affect the accuracy of date rape drug detectors.
To conduct this experiment, 7 different non-alcoholic and alcoholic beverages were used. These liquids were selected due to their common prevalence in settings where date rape crimes occur most often, such as bars, clubs, or parties. The non-alcoholic beverages used were filtered water, flavoring sparkling water, and soda. The alcoholic beverages used were beer, alcoholic seltzer, tequila, and vodka. Ketamine in a pure powder form was used as our independent variable. Although the test strips and coasters advertise that they can detect GHB as well, we were not able to obtain this drug because of its classification as a Schedule I drug.
The test detectors used in this evaluation were What's in Your Glass coasters by DrinkSafe Technologies and What's in Your Drink test strips by Test My Drink. Users are instructed to place a small drop of the drink onto each of the circles of the test and wait until it is fully dry to view results. If either circle noticeably changes to a darker blue color, this indicates a positive result, meaning the beverage is spiked. If there is not a change in color or only a slight change in color, this indicates a negative result.
For our methods, we tested the test strips and coasters individually using a control group with no ketamine added and an experimental group with ketamine added. As described in the detectors, realistic concentrations are considered to be 1 gram of ketamine per 250 milliliters. These same concentrations were used in this study, but were scaled down due to limited amounts of ketamine available for experimental use. We tested coasters and test strips in 3 categories: below realistic concentrations, at realistic concentrations, and above realistic concentrations according to package instructions. We repeated this process for a total of 236 individual coaster detectors and 236 test strip detectors.
We consider detectors to be accurate if they correctly identified that there was ketamine in the samples by turning a significant blue color. It should be kept in mind that we found detectors are 100% accurate when ketamine is not present in the liquid. This graph shows the average accuracy of coaster and test strip detectors at the 3 different concentrations of ketamine in liquids. A low concentration is considered to be a 0.5 gram per 250 milliliters. 1 gram per 250 milliliters is considered to be the normal or realistic concentration, and 1.5 grams per 250 milliliters is considered to be overconcentrated. The most important percentages are those in the middle, as this shows the accuracy at 1 gram of ketamine per 250 milliliters, which is what the drug testing kits deem as realistic concentrations. For realistic concentrations, test strips are 93% accurate and coasters are 77% accurate. This graph also shows that as the amount of ketamine concentrated in liquid increases, the accuracy of the tests also increases. This graph shows the accuracy of coasters and test strips divided by liquid type using realistic concentrations of ketamine only. Overall, test strips appeared to have a greater accuracy than the coasters, with the outlier being sparkling water, which has a 67% accuracy with the test strips and an 83% accuracy with the coasters. Another outlying result is that the coasters only indicated a positive result 50% of the time when using beer, but the test strips with beer were 90% accurate, which is a large difference, and most of the coasters and test strips of the same liquid were closer in accuracy than this.
Based on our data, 2 out of our 3 hypotheses were correct. Our first hypothesis was incorrect. We hypothesized that coasters and test strips would have the same accuracy. After our experiment, however, we saw that test strips were performing better than coasters overall, with test strips being 16% more accurate than coasters at realistic concentrations, and test strips being 13% more accurate than coasters at above realistic concentrations. Statistical analysis calculated a p-value of 0.005, which was less than our significance level of 0.05. This means that our hypothesis was incorrect because there is significant evidence that test strips are more accurate than coasters. Our second hypothesis was correct. We found that change in concentration of ketamine does affect the accuracy of date rape drug detectors. In our experiment, as the concentration of ketamine increased, so did the accuracy of the detectors. Our p-value of 0.00017 was below our significance level of 0.05. So we can conclude that there is significant evidence that changing the concentration of ketamine in the beverage will affect the accuracy. Past research had not determined the effects that changing concentration of ketamine has on the accuracy of date rape drug detectors, so this finding is new information. Our third hypothesis was also correct. Our data supports that a change in liquid does not affect the accuracy of date rape drug detectors. We did find that there were outliers in our data, but these outliers were not considered significant because our statistical analysis calculated a p-value of 1.01, which was greater than our significance level of 0.05. This means that we can conclude that there is no significant evidence that accuracy is affected by liquid at realistic concentrations of ketamine.
The study was not only conducted to determine accuracy, but also efficacy in terms of how these detectors would be realistically implemented if a person was not in a lab but in a bar or party setting. First, for all of our findings, with no ketamine present in the beverage, test strips and coasters are both 100% accurate. This draws the conclusion that these detectors will not create false positives, but their inaccuracies when ketamine is added mean that they can produce false negatives. False negatives are a bigger problem than false positives because false negatives mean that the customer will most likely ingest a spiked drink. Also, the limits of detection are concerning as the detectors only work in realistic or over-realistic concentrations, meaning that there has to be at least 1 gram of ketamine for every 250 milliliters of liquid. There is some threshold between half a gram and 1 gram of ketamine where the test strips and coasters start to accurately detect the presence of ketamine within a beverage, but this concentration is much higher than average doses. The average doses of ketamine that a perpetrator would use are usually between 0.075 to 0.3 grams per 250 milliliters of liquid. The lowest concentration of ketamine in each beverage used in our study was half a gram per 250 milliliters, which is significantly higher than average doses. This is concerning because at the lowest concentration we used in our study, the detectors were 0% accurate with the presence of ketamine. This data suggests that they will also be 0% accurate at concentrations lower than half a gram per 250 milliliters, meaning the detectors would not pick up the average dose of ketamine used in drug-facilitated date rape. Another issue with the products is that the detectors only test for ketamine and GHB, but there are many other drugs that a person may use for drug-facilitated rape, such as MDMA, Ropanol, Xanax, and others. Just because a test may say a person's drink is not spiked, it could be spiked with a drug that the tests are unable to detect. The third issue with the efficacy of our detectors is that the instructions are kind of unclear. When using the detectors, directions on the package only notify the consumer to, quote, place a drop of your beverage onto both spots of one test, unquote. These instructions are vague and leave room for error, as using too much liquid could produce an inaccurate result. Also, many people would not know how long to wait for the test, as the directions simply say to wait until dry. A consumer may falsely believe that they are safe because their test result is negative, but in reality they may not have waited long enough for the test to dry. This is because the ketamine needs to be fully evaporated before the cobalt thiocyanate will turn a darker blue color. In addition, the consumer may falsely interpret the test results because of darker lighting and the subjectivity of the phrase a darker blue color, especially because accurate positive tests often display a fainter blue color than one might expect. In the photo above, there are 2 tests with a concentration of 1 gram of ketamine per 250 milliliters and 2 tests with no ketamine. The coaster and test strip on the right were deemed inaccurate for producing a false negative result, as they either barely showed or didn't show a dark blue color. This demonstrates the faults of these tests and how dark lighting could affect the use of these detectors.
Our results find that date rape drug detectors are not acceptable in realistic situations. While test strips may be more accurate than coasters, this does not mean that test strips should be used over coasters. Accuracy is unreliable, especially at lower concentrations of ketamine, and efficacy of using these products in realistic settings is not acceptable. Therefore, it is clear that increased education needs to be provided about other preventative methods surrounding drug-facilitated date rape. We recommend that neither coasters nor test strips are used solely as a safety resource to detect date rape drugs and beverages, because their inaccuracies, especially due to false negatives, can create a false sense of security and put the consumer at risk.
The experiment was controlled by using several alcoholic and non-alcoholic drink varieties, a powder form of ketamine, and different concentrations of ketamine for each trial. Even though the environment was controlled as much as possible, there were still limitations of the study that may have affected the results. First, our experiment was conducted under lab conditions, which influenced the lighting, atmosphere, and our alertness, all of which could be very different in a real-life situation. Second, we could only obtain access to ketamine, so we were unable to test for GHB. And finally, the instructions on the products are fairly subjective, so our interpretation of a darker blue color could be different than someone else's interpretation.
In the future, researchers may want to ask if there are other ways to make detectors using technology that is not affected by outside variables. This would allow for future detectors to be more accurate and have fewer limitations around how they can be used. It would be even more beneficial to research the root of this problem, such as the psychology behind date rape drug perpetrators. This solution would be favorable because it would not put the matter into the hands of the victim. Instead, it would put the responsibility into the hands of the perpetrators. This would only occur through a change in societal behavior, not chemistry. Overall, more options should be provided on how people can stay safe from drug-facilitated date rape, considering that these detectors are not accurate.
We want to give a special thanks to Nick Wyatt and Harbor Health and Apothecary for making this project possible by giving us access to a laboratory and ketamine, which we would not have been able to obtain otherwise. Special thanks to Mr. DeGroat for facilitating our project by helping us edit and refine our paper and keeping us on track. Also to our parents for constantly supporting us, and of course the Marine Chem class of 2022.
Here are our sources. Are there any questions? So the question was, which test for significance did we use? And we used a chi-square test for the accuracy of the test strips in the coasters based on the change in liquid and the change in concentration. And we used a 2-proportional z-test for if it was significantly different between the accuracy of the coasters and the test strips. The question was, how much do the products cost? You most of the time can buy these products in bulk, and we would estimate that the products cost under $2 each.
The question was, have we considered letting the manufacturers know that like the tests don't work in realistic settings? And yes, I think that would be a really good idea to let them know that. But there are also other studies that say that these work better than like ours, but they're also done at the concentrations that they say on the instructions only, so that would definitely be a good thing for us to let them know.
Could you repeat that again?
The question was, how different would the results be at a bar or club, like in a real life situation? And I think that the accuracy of the tests would be the same as what we found, but I just forgot what it was.
At realistic settings, I think lighting would be a big factor, because in a bar or a club, there might be disco lights or it might be darker lighting. And as we showed in one of the slides, it already is hard to tell if there is a significant blue color. So I think lighting would change, and I think user error could affect and make the tests less accurate, because if a person does not wait long enough or they put too much of their beverage on the test, that could also decrease the accuracy. So overall, I would say that I think the accuracy would decrease in realistic settings.
Test. Hi, my name is Drake Dallempines, and my project was on the quantification of microplastics from tributaries to the Puyallup River. So, what exactly are microplastics? As the name might suggest, microplastics are quite small and are defined by the National Oceanic and Atmospheric Administration as any piece of plastic less than 5 millimeters in size. This photo shows a blue microplastic in a plankton sample. Microplastics are divided into two categories based on how they form. Primary microplastics are shed from commercial products such as clothing, tires, and cosmetics, while secondary microplastics are fragments of larger plastic pieces that are broken down due to natural erosion from the wind, waves, or sunlight exposure. Primary microplastics come in several configurations, which include microbeads, microfibers, and nurdles, which are plastic pellets manufactured and melted down to create other plastic pieces ranging from bags to car parts. This is an example of secondary microplastics, and as you can see, they are much more irregular in shape than their primary counterparts. Microplastics are everywhere and are a major threat to the environment. Their extremely small size makes them difficult to contain. This often leads to their consumption by animals on all levels of the food chain. Since they are indigestible, microplastics clog the stomachs of marine organisms and can lead to their starvation. If not addressed, this could lead to human consumption of microplastics as they make their way up the food chain. This can be seen in this diagram, where a bottle thrown into the ocean breaks apart into smaller plastic fragments that are then consumed by fish, which can wind up being eaten by humans. It is thus extremely important to try and minimize the amount of plastics that end up in our oceans. One way to achieve that is by quantifying how much plastic enters through rivers flowing out to sea. This will allow scientists to devise strategies to address problem areas with regards to preventing microplastic spread. An alarming 4.8 to 12.7 million tons of plastic ends up in the ocean each year. In the state of Washington, which has a huge maritime industry and is second in the nation for seafood processing, microplastics entering the food chain through organisms that are essential to the state's seafood operations are a definite problem. The impact is not limited to aquatic creatures and many other animals, such as seabirds, tragically mistake plastic for food. The Puyallup watershed is comprised of a group of rivers, with the main one being the Puyallup itself, that drain from the highly elevated Mount Rainier National Park down and out over 46 miles to Commencement Bay. It is divided into two parts, with the upper Puyallup covering the majority of the watershed from Mount Rainier down to Orting, while the lower Puyallup, the area tested in my study, ranges past Orting all the way into Commencement Bay. Previous research conducted by Alstead in 2020 had analyzed 6 sites along the Puyallup in the lower part of the watershed. His study looked at microplastic concentration relating to the urbanization of surrounding areas. This in turn was heavily inspired by the work of Masura and a team at the University of Washington, who tested the same area and focused on microplastic concentration in relation to wastewater treatment. Washington's rivers as a whole have not had much research conducted on them regarding microplastic concentrations, and with 11 of Washington's major rivers flowing into the Puget Sound, it is important to try and quantify their individual microplastic contributions to the Salish Sea. In 2020, Alstead concluded that there were indeed many microplastics adrift in the Puyallup and that there was some correlation between an area's industrialization and the amount of microplastics that entered the Puyallup River. With my project, I took this a step further by looking at how several tributaries to the Puyallup affected this total count. My study therefore looked to elaborate by sampling 7 different tributaries to the lower Puyallup River, as well as the mouth of Commencement Bay. My sample sites ranged from around Orting down to the mouth of the Puyallup. Sites were chosen based on how close they were to where each tributary intersected with the Puyallup River and also took accessibility into account. However, I was unable to access an 8th site at Canyon Falls Creek due to it being on private property. My hypothesis was that the tributary to the Puyallup River that would yield the most microplastics would be the White River due to the abundance of commercial establishments in the surrounding area of the sample site. The independent variable in my study was the specific tributary I took a sample from, while the dependent variable was the amount of microplastics present in each tributary. One of my many control variables was that the samples were taken on the same day in the same weather conditions. I initially planned to use a manta tow to collect my samples, a device that is deployable in running water and generally collects a larger sample size. The Bellarmine Marine Chemistry Lab actually possesses a manta tow, but while it would have yielded the best water samples, it would have been too cumbersome to deploy in smaller creeks that are not as wide or deep as the larger bodies of water that other researchers tested. Instead, I used relatively cheap and accessible equipment in order to make this study more replicable. Therefore, I chose to use a bucket for sample collection. This was made of metal to avoid potential plastic contamination. This bucket was tied onto a roped pole, which was then lowered into each tributary I sampled. These pictures depict my sample collection technique at several of the tributaries. At each site, the bucket was filled and emptied thrice with each tributary's water to ensure sample quality and prevent initial contamination before collecting the sample. At tributaries with access points that exceeded the range of the pole, the bucket was tied to a 100-foot rope instead, which was lowered and raised manually. This picture shows me collecting a sample from the White River using the rope in place of the pole. After sample water was collected with the bucket, samples were then stored in glass jars to prevent undesirable microplastic contamination from outside sources. 35 total samples were obtained, with 5 derived from each site. After all samples were collected, they were stored in the Bellarmine Marine Chemistry Lab. Each jar had a maximum volume of 350 milliliters. Water was then removed from each sample until 250 milliliters remained. This was done to ensure uniform microplastic concentration per unit volume. First, samples were sequentially filtered through 3 sieves measuring 5 millimeters, 1 millimeter, and 330 micrometers. Samples were then dried in an incubator at a temperature of 50 degrees Celsius for 2 days, and from there were treated with 3 drops of bleach in order to break down any organic material, as bleach is known to dissolve skin cells and other biotic matter. Samples were then placed onto microscope slides, and cover slips were applied to each. Samples were counted in a directional manner from top to bottom and left to right until the entire sample had been viewed. Because microplastic appearance can vary significantly, I chose specific criteria for their identification. These included clear absence of a cell wall in chloroplasts, lack of other organelles, split ends of fibers, unnatural shapes, and vibrant artificial colors. While I had predicted that the White River would have the highest average, it was only second highest among the tributaries and third highest when including the sample taken from the mouth of the Puyallup. The tributary that ended up having the highest amount of microplastics per sample was Swan Creek, with an average of 28.4 microplastics. In sharp contrast, Clear Creek, living up to its name, had the lowest amount of microplastics per sample with an average of 5.4. I plotted the average microplastic count in relation to the surrounding population density of each sample site. As you can see from this gradient bar, a darker color indicates a higher population density measured in people per square mile. A small creek, which I dubbed the Mini Puyallup River, had the highest surrounding population density. However, it had the second to the lowest prevalence of microplastics. Therefore, other reasons must be entertained when considering contribution of microplastics to nearby bodies of water. Swan Creek, the tributary with the highest microplastic count, only had a moderate population around it. However, the presence of a tire shop nearby may have contributed to its relatively higher count. As we have seen, tires are a significant and often overlooked source of primary microplastics, as several studies have also examined. Additionally, the amount of vehicular traffic, and by extension tire wear, may also have contributed to the individual microplastic counts, something that may be worthwhile for a future study. In fact, a study done by student designers in the UK regarding the capture of microplastics flying off of car tires found that microplastics shed from tires comprise approximately 550,000 tons of the plastic that pollutes the oceans. Overall, my hypothesis was incorrect, but still partially supported due to the White River having the second highest microplastic total among the tributaries. Some limitations of the study were possible observer error when counting and identifying the microplastics, as well as other confounding variables not directly observed. Future research could potentially investigate ways to prevent the release of microplastics into the environment, as even in low-populated areas like Swan Creek, there was much more plastic overall than more densely populated areas like the Mini Puyallup River. In addition, further research needs to be done on other Washington rivers, such as the Snohomish, and researchers could look to compare these numbers with those of the Puyallup. Potentially looking at the upper Puyallup watershed could also bring further clarity to how polluted our rivers, and specifically the Puyallup, are. Finally, fish sampling may be useful in determining the extent of microplastic uptake by wildlife, especially those that are sources of food. Perhaps one day, research such as this can turn the tide in favor of our precious wildlife that has suffered for far too long. I'd like to thank my family for their love and support, and especially my dad for helping me to actually collect the samples at each site. Thank you to Mr. DeGroat for guiding me along every step of the way and assisting me with the research and presentation process, and also for being the greatest van driver. Huge thank you to Asa Alstead, who inspired the basis for my project and gave me helpful tips and information regarding sample collection. Finally, shout out to the Marine Chem class of 2022, especially the greatest van group from TSN 292. It's been an awesome 4 years with you guys. These are my references. Any questions? Grace. Okay, so the question was why I hypothesized that the White River would have the highest microplastic count, and the answer to that is because there are lots of commercial establishments, restaurants, gyms, even a library around where I took my sample. So I thought this would be cause for a higher overall microplastic count for that river.
Are there potential other variables about how the microplastics maybe exist in the water or flow through, like depth of the sample or near the shore or any of that kind of stuff?
Okay, so the question was whether the height or depth that the sample was collected at may have had other variables when determining the microplastic count. And I think it definitely had something to do with that, and that could be an area of future study by looking at maybe different distances from the shore, maybe different depths of actually collecting the water sample from the surface. Could you repeat that? Sorry. Okay, so the question was, how will the information I've collected impact the people who live along these rivers? And I think that it's important, especially for those who live along creeks such as the Swan Creek, which has a residential neighborhood a little upstream of it. I think it's important for people to monitor how much single-use plastic they use and try to use more sustainable sources. And yeah. Thank you.
Hi, my name is Briaya,
And my name is Haley Dillow.
and our project is determining the presence of Phytophthora species in soils of diseased alder trees at Titlow Park. While walking around Titlow, we noticed an abundance of diseased alder trees around the trails and nearby waterways. Curious as to what could be causing the poor health of these trees and our initial research, our initial interest in environmental science, we decided to start our research here. In taking a closer look at Titlow, we noticed a ton of dead alders with bleeds on the bark and bare branches, hence why we decided to collect soil around these trees. Titlow has been an important research site for Bellarmine and marine chemistry program for countless of years. Titlow provides tons of things like walking trails, summer camps for kids, scuba diving, dog walking, and children's parks. On top of that, alder trees in particular are key to nitrogen fixation, which is essential to life because fixed inorganic nitrogen compounds are required to biosynthesize basic building blocks of plants, animals, and other life forms. Alders also provide habitat for terrestrial and aquatic organisms, provide shade and cooler temperature conditions, and help stabilize riverbanks. Studies have shown that forests containing a large amount of alders have a higher biodiversity value. This is because the dense shrubbery associated with alders houses many different wildlife species. In addition, it's a pioneer species, meaning it aids in natural repair and colonization if an area is disturbed from a natural event like a landslide or flooding. Once an alder tree dies, it is difficult for them to regenerate themselves because they can be selective about their soil conditions. This is why it's necessary to specifically investigate the soil conditions of the park. The protection of these trees will ultimately save the delicate ecosystems present at Titlow. In a previous study done in 2016, there was found a substantial decline in alder tree health along streams in Europe because of an invasive species called Phytophthora olney. Considering Titlow's alders are found in an aquatic environment, we assumed a type of Phytophthora similar to Phytophthora olney was present because of the similarity of the ecosystems. If alder trees are being affected by Phytophthora, other plants in close proximity are also being affected. Phytophthora is very transmittable because it contains swimming spores called zoospores that can swim through films of water in saturated soils to locate new host plants. If an alder tree is getting its infected water from the soil around it, that same water is being taken up from other plants. If not controlled, an entire area could be wiped out from Phytophthora. This could be devastating for Titlow's environment.
So, what exactly is Phytophthora? Phytophthora is a highly transmittable plant-killing omocyte or water mold, similar to brown algae. Phytophthora is a concerning invasive species affecting forests as well as nurseries all over the world, and when present, often forms bleeds on the outer bark of trees, similar to the ones we observed. Preliminary results researched by Marianne Elliott of Washington State University found Phytophthora in alders in nurseries and was concerned about its spread. Non-native forest pests and pathogens like Phytophthora negatively affect the health of forest ecosystems. Once Phytophthora is introduced to new environments, these organisms can change forest community structure and species composition, altering ecosystem function and offsetting the co-evolved natural balance and the overall resiliency to environmental changes. The damage caused by invasive pests and pathogens also costs taxpayers billions of dollars annually for environmental repair. Preventing or intensely managing these invasions is critical for protecting the benefits forests provide to society.
This image above is an example of what a Phytophthora spar looks like and is actually a picture we took ourselves from one of our samples later in the study. So the main question we explore with our project is, are there species of Phytophthora present in alder soil that could be contributing to the rising number of alder tree deaths at Titlow Park? And if so, which species? Throughout the course of our project, we have to keep in mind that no matter what we find, we cannot directly state that X species is causing the death of alders at this location. We can only draw connections between tree health and the species we find to give future researchers insight on the patterns of specific species of Phytophthora.
Based off of our initial observations, we hypothesized that because of the canopy dieback and Phytophthora bleeds present on alder trees, there is at least one species of Phytophthora affecting Titlow's environment. Our independent variable for this study is tree health, specifically the dieback percentage of each alder tree and from which site it came. We intentionally varied the soil samples we took from trees based on variation in dieback so that we'd have a range from fully healthy to completely dead alders at each of the 4 sites. And our dependent variable is the varying Phytophthora species that could be found at differing health levels and differing sites.
So this is a map of the specific trails in Titlow Park and the location of our 4 sites relative to each other. We selected sites that were around a quarter of a mile away from each other to ensure an accurate representation of alder tree health across the entire park. The parameters for each site was that there was at least 8 alders per location, with a range from completely dead to completely healthy trees. The health of each tree was determined by canopy dieback. A tree with 0 canopy dieback would have an abundance of projections and leaves, whereas a tree with 10% canopy dieback would have no leaves or very few projections coming from the trunk of the tree. The labeled dieback percentages in the picture above are roughly what we used in determining the dieback percentages of our trees.
Our method started with the field work, which was locating the sites, estimating the canopy dieback, and then the soil collection. To start collection, we chose the 4 sites around Titlow, following the circular trail loop, with each site having close to a 50-foot radius. From there, we chose 4 alder trees per site, trying to get a range of both healthy and dead trees like mentioned before. To start soil collection, we scraped off the top layer of moss and leaves to prevent contamination, and then dug 2 to 5 inches deep into the soil from 3 different locations around the base of a tree to collect a total of half a liter to a liter of dirt per alder. Once all these 16 samples were collected, at the WSU lab, we put 100 grams of soil per tree into these bottles and flooded them with purified water. 2 healthy rhododendron leaves were then put on top, and the bottles were laid on their side for 5 days. We used rhododendron leaves because they were previously being used in a WSU study and were already found to have no other diseases or mites on them. Doing this gave the potential Phytophthora time to swim to the leaves and infect them, something known as Phytophthora baiting. If Phytophthora is present in water, zoospores will swim towards the surface of the water surrounding the bait, colonize it, and develop dark brown lesions. In this case, the rhododendron leaves were our bait. If the tested water is contaminated with a high amount of the pathogen, those brown lesions will appear within 2 to 3 days after the baits are suspended in the water, hence why we left the bottles for 5 days, to give the potential Phytophthora time to swim to the leaves and infect them. Once the leaves were removed, 10 hole punches of these spores were isolated per tree. These isolates were then plated onto petri dishes using sterilized tweezers. So after plating these isolates, the petri plates were left in a drawer for a week-long incubation period. Once removed, they looked something like this. Many of the isolates had growth surrounding them. Phytophthora specifically has a circular defined outline. So you can see from these 4 isolates, it all has a defined ridge. But this one down here didn't. It had projections growing instead of a defined circular shape. This is an indicator for a fungi called Pythium, which something almost all plants are susceptible to, but it wasn't specifically Phytophthora, which is what we were looking for. So from these original plates, new isolates were removed from only the specific Phytophthora cultures, which you can see these little boxes. And then these were replated and sent out for DNA sequencing.
Okay, so to better understand our process, it's important to do a little overview of what PCR actually is. First off, it's nearly impossible to analyze a single isolated sample of DNA. To accurately accomplish this, you must either have a very large sample of DNA or go through a process known as polymerase chain reaction, or better known as PCR. This process is sometimes called molecular photocopying for the very reason that this process makes many copies of the sample provided to create enough DNA for analysis. So from our samples in particular, to produce this reaction, we needed a sample of our DNA, DNA primers, which promote synthesis of complementary nucleotide strands, and DNA polymerase, which is an enzyme that allows for the synthesis of a complementary strand of DNA. And finally, a nucleotide mixture of the ATGC base pairs to physically duplicate the strand we're copying. This specific segment of DNA we analyzed in each sample is known as the ITS region, which stands for internal transcript spacer and is the universal region for differentiating fungi. The sequence of the ITS region usually possesses more variation than that of the ribosomal sequence, making it an ideal region for analysis. The product of the PCR is then sent to another lab that has access to Sanger sequencing equipment to then provide a detailed DNA sequence of the sample. This process is of course repeated for each specific sample collected from our Petri plates. Okay. And the newly amplified DNA is then ran through a program called BLAST, which stands for Basic Local Alignment Search Tool, where the DNA code is entered and matched to an online database of other sequences to identify the species of our specific sample. For our data, we used the BLAST NCBI GenBank. For example, Phytophthora pseudosyringae was a 98% match with our sequence. The image above is an example of the DNA base pair sequencing of a single sample of Phytophthora found in our collections. And up close, we can see the individual base pairs.
So here's two tables that pretty much summarize our results. Out of all of our samples, we were able to detect 3 species of Phytophthora. Phytophthora cambivora was found in only alive trees, while Phytophthora siscuensis was found only in dead trees, and our third species, Phytophthora pseudosyringae, was found in both dead and alive alder trees. The presence of siscuensis was found in sites 1 and 2, which had the most canopy dieback except for site 4, which seems to have had something else affecting the health of the trees. The first table shows each site, its average canopy dieback, the presence of dead trees, and the types of Phytophthora found, which is all pretty much summed up into table 2, which more directly states which species were found according to the health of the specific tree that species was isolated from. So for Phytophthora cambivora, for example, even though it says yes to dead trees present right here, the species itself was only isolated from one tree, which happened to be one of the healthy trees at that site.
Moving into our discussions, Phytophthora pseudosyringae was found at all sites in trees of healthy and dead alders, which may suggest this species is not causing severe harm. Because it was found in all 4 sites, it is possible it spread from people on trails. Phytophthora siscuensis was found at 2 sites and only recovered from 2 dead trees. Being that this species was found only in dead trees suggests that this Phytophthora species is more harmful than the other 2. And finally, Phytophthora cambivora was found in only one of our trees that was healthy. So it is possible that Phytophthora cambivora was recently introduced to Titlow because we did not find it as frequently as the other species.
We have a couple of recommendations for Titlow in both dealing with and preventing their Phytophthora problem. Besides being spread through water and soil naturally, Phytophthora can spread from place to place by people. Reducing the walking off of trails is imperative in preventing the spread of Phytophthora from one location to a different one around the park. Because we know Phytophthora is already present, using hot steam to sterilize soil and dead plants can stop the spread of Phytophthora from one plant to a nearby one, which could slow the spread of Phytophthora across a specific area. And here's a picture of that. Improving soil drainage decreases the chance of an abundance of Phytophthora swimming near the tree roots and infecting the plants nearby. And then lastly, for a more expensive treatment, around $200 per tree, trees can be injected with phosphonates that blocks the zoospores from entering the plant tissue, which has been shown to help trees resist Phytophthora and its negative effects.
Throughout our research project, we encountered a few limitations. Our first one is that our soil collection was during the end of summer, which may have excluded the possibility of different species of Phytophthora being dormant, waiting for the right conditions such as warmer weather during the midsummer or being more easily transmitted during the rainy seasons of spring and winter. And another potential limitation of our study was cross-contamination of the samples. It is possible the shovel we used while rinsed in between each sample wasn't fully decontaminated. This could have affected where Phytophthora was supposedly found.
More research is needed to confirm that these Phytophthora species are in fact killing the alder, because other organisms could be involved. Specifically in Site 4, like I said, Phytophthora was not abundantly recovered, but instead these trees had a lot of insect holes and there was a presence of a fungus called Armorillia. Our experiment wasn't testing correlation between Phytophthora and tree health, but more just testing for identification of species. We suggest in a controlled environment to study the correlation between Phytophthora and alder tree death by infecting healthy alders with the specific 3 species that we found to prove that that is the sole cause of the alder tree death that we've been noticing at Titlow. And doing this can also help compare the harmfulness of the 3 species that we found. Another future study could experiment with the connections between the spread of Phytophthora and a tree's proximity to either water or the trails. And lastly, instead of using collection from the soil around the trees like we did, future research could use canopy collection or collect from the water at Titlow for projects similar to ours, testing for Phytophthora identification.
Okay. So we want to thank Dr. Joey Holbert.
No, so Phytophthora affects, here we go. Phytophthora affects almost all plants. But the reason we chose alder trees specifically, because we mentioned in the beginning just how important they are for nitrogen fixation, habitat, for especially for Titlow. So that's why we specifically chose alder trees, but Phytophthora affects almost every plant.
Mina. Okay, so the question was, what sparked our interest in this topic? So we came in contact with our mentor through Mr. DeGroat, and he had actually done a previous study similar to this one, but his paper was never published, and he wanted to do a continuation that involved some of the variables he wasn't able to include in his past study.
Also, to go back to your question, his previous research was on lavender. So we actually decided to just do something a little different, just to see if, things were the same between lavender and alder.
Wait, we have one more question.
Yes. Yes, you. So the question is, the soil steaming that I showed, is that being done at Titlow? As I'm aware of right now, that is not being done at Titlow. I think that's more used in controlled, like nursery environments. It's a little easier to do that there or for irrigation, but I think it would be a good idea for Titlow to implement in order to kind of correct their Phytophthora problem.
The question was if we knew if Metro Parks was even aware of this problem. I'm assuming not, honestly, just because Phytophthora is actually a widely unknown problem, even though it does cost billions of dollars, worldwide. So it would be a good idea to let Metro Parks know. So I'm sure we'll do that.
Our mentor, too, specifically noticed a lot of dead alders. So I think that's becoming kind of abundant, is this all of a sudden rapid decline in alder tree health. But I don't think people are aware that it's Phytophthora necessarily causing that problem.
Why don't you go ahead, Joe?
How do I hold it? Okay. I'll figure it out. Here? These?
Yeah.
Okay. Yeah. There you go. Yeah. Hello? Okay. Sorry. Good evening, everyone. My name is Grace Turner, and my project is on testing the effectiveness of aeroponics versus traditional growing methods on Lactica sativa in the Bellarmine greenhouse. So what is aeroponics? As defined by Oswald in his 2001 study, aeroponics is a form of plant growth exempt of soil where the roots are suspended using artificial support. It combines air and water, and the roots hang in darkness within a sealed container underneath the lid. The roots are constantly exposed to oxygen and sprayed with nutrient water using pressure nozzles to encourage rapid growth and maintain controlled conditions. Nur added to this in his 1982 study by defining that the top of the plant should be excluded from the wet zone as well. The first instance of air plant growing systems was in the early 1920s. Barker's 1922 study found that a lack of soil was preferable, and Carter added to this in his 1942 study by cultivating pineapples successfully through the air growing method. So why aeroponics? Why not hydroponics or aquaponics? Aeroponics has fared well against hydroponics and aquaponics, saving more water and ultimately producing higher yields. Wendt's 1957 study grew tomatoes and coffee in watertight containers with nutrient mist, and Buer added to this in his 1996 study with the nutrient mist method, and he found that this was optimal for environmental growth but minimal water use. Eventually, in NASA's 2006 study, aeroponics was discovered to save up to 99% of water, 50% of nutrients, and was 45% faster than cultivation using soil. This study was especially relevant to my interest in aeroponics and its application for the school because it proved it to be a sustainable method of growth. So Bellarmine Preparatory School, as a Catholic institution, follows many Jesuit principles, one of which being to care for our common home. This call promotes environmental consciousness and stewardship. In order to achieve this principle, Bellarmine has initiated several efforts towards sustainability. Most notably, Bellarmine's environmental sustainability representatives have begun recycling and composting initiatives, but additionally, Bellarmine constructed the E.L. Wigan greenhouse in 1996 for the scientific study dedicated to the growing and research of native and endangered plant species. Most recently, Wetzel, a previous marine chemistry student, in her 2021 study, utilized the greenhouse to compare hydroponic tomatoes to traditionally grown tomatoes, a project very similar to mine. Her results indicated that the greenhouse provided stable enough conditions for the tomatoes inside to grow successfully. So to contribute to Bellarmine's efforts, I focused on utilizing the school's greenhouse to combine aeroponic research with sustainability. So the research surrounding aeroponics demonstrates it as a sustainable method of growth. Aeroponics uses less water but continues to result in healthy vegetation. My independent variable in this experiment was my aeroponic system, and my dependent variable was butter crunch loose leaf lettuce. So in the following experiment, I sought to prove the effectiveness of aeroponics with butter crunch loose leaf lettuce under Bellarmine Preparatory School's greenhouse conditions. I hypothesized that the growth rate of aeroponic lettuce would be faster than that of the control, and I also predicted that the amount of water used in the aeroponic system would be less than that used for the control group. So to begin with my methods, using one 17-gallon tote as the vessel for my aeroponic system, I purchased 10 feet of PVC piping and cut this into multiple sections that connected to a pond pump. So in the image on the right, this pump located at the bottom is what fueled my system, and this piping along the perimeter is where the water came out of. Additionally, I cut small holes into the PVC piping that was measured around the perimeter, and 180-degree and 360-degree nozzles were inserted into this. So my 360-degree nozzles were these green nozzles, and the 180 degrees were the red ones. Lastly, to finish off the building of the system, I cut 9 holes into the top of the lid, and this was where my net cups would be inserted into. This system was modeled after Zobel and Mirza's design, but I simplified it for cost efficiency. Next, the plant chosen for the study was butter crunch loose leaf lettuce, as I mentioned before, scientifically known as Lactica sativa, because it's a loose leaf lettuce which grows fastest compared to other lettuce variants. I compared butter crunch lettuce to salad bowl lettuce when I initially planted my seeds, and I found that the butter crunch grew faster and taller, so I chose this for my study. And after being planted, my seeds were sprayed with water once every day for 20 days, and 2 lamps were placed to the growing seeds until they were transplanted for 12 hours each day. So the following image is of my control group. After 20 days, the seedlings were transported to Bellarmine, and 18 butter crunch seedlings were chosen to be used for my study. 9 seedlings were chosen to be transplanted into net cups, and 9 were chosen to be inserted into the control group, larger containers for the control. So I dug out each seedling from a seed starting tray that I started my seeds in, and carefully dug a hole into my control pots and placed the seedlings there. And clay pebbles were used to stabilize the seedlings which were moved into net cups, mostly because these clay pebbles don't have any extra added nutrients, so it wouldn't be a confounding variable. Finally, gardening training wire was spiraled around both groups to help stand them up and in an upright position, and this is to help me with measurements. So this is the interior view of the aeroponic system without the piping or the pump. And as you can see, it's filled up with water, and this white powdery substance at the bottom is the nutrient mix. So the aeroponic system tote was filled with 5.5 gallons of water initially, and 10 teaspoons of hydroponic nutrient mix was added, which in this image I'm mixing in. The aeroponic lettuce seedlings receive sprays of water for 60 seconds every 24 hours using an aeroponic timer which would regulate the watering cycles. The control group was watered using a 50 milliliter graduated cylinder, and the amount of water was measured and adjusted for the control group as needed. I used a very practical and simple gardening technique of placing my finger into the soil about an inch deep and taking it out. I would observe how much soil was sticking to my finger to determine whether my plants needed more water, or were getting too much water, or were getting an ideal amount of water. So the reservoir for the aeroponic system was measured after 1 month of measurements, and the seedlings were exposed to a heater to accommodate for some of the colder greenhouse conditions, considering I did this project in the winter. And this image is an exterior view of the aeroponic system. As you can see, the lettuce seedlings are those green plants growing up at the top. The clay pebbles are those brown little pebbles, and those are the net cups. The roots will hang down inside the tote. And then the dark green next to the seedlings is the training wire that was meant to help keep them in an upright position. So the height of the lettuce was measured in millimeters for the aeroponic and control group using a standard ruler after being transplanted. So these are my results. This graph depicts the height of the control group lettuce over time. So my x-axis is the date of measurement, and my y-axis is the height of the lettuce in millimeters. So each individual colored line represents one of the 9 lettuce controls, and each dot represents a day that it was measured. So as you can see from the graph, the control lettuce had overall steady and gradual growth, and early on, as I'll point out here, certain lettuce did actually experience some shrinking, which I'll address later. This graph depicts the height of aeroponic lettuce over time, with the X and Y axis the same as the first graph. Again, each line represents an individual lettuce, but this time in the aeroponic group, and each dot represents a day that it was measured. So as evident, the aeroponic lettuce experienced more significant growth and did not experience any shrinking. As you can see, the dates are the dots, and the lines just continue to go up, none of them dip down. So this is my final graph, and the following graph demonstrates the difference in growth rate between my control group, which is the blue line, and my aeroponic group, which is the orange. So as you can see, the orange line always remained above the blue line, and the control group even experienced some negative growth early on. So as I mentioned in my control graph, they shrunk a bit in the beginning, and this is really evident in the beginning of this graph, experiencing some negative growth. The spikes of growth were notably higher for the aeroponic group than the control group, and they were more frequent. And my last piece of data was water usage. So the amount of water lost from the aeroponic system was measured to be 998 milliliters, and the amount of water used by the control group overall was 1,130 milliliters. So the following experiment was actually my second trial comparing an aeroponic group to a control group. In my first trial, I used salad bowl lettuce instead of butter crunch lettuce, and the seedlings were transplanted much earlier. I ended up transplanting the first trial of seedlings after only 3 days. They were very weak, they were very thin. My timer interval for the first trial was 15 seconds of water every 5 minutes, continuously every day. So my aeroponic lettuce as well as my control group overwatered quickly, turning brown and shrinking. As you can see on the image on the right, the lettuce is long but turning brown and decaying. No height measurements were collected from this first trial, but the mistakes that I made led to significant changes in my methodology for the second trial. And from the second trial, my results confirmed my hypothesis that the aeroponic group would grow faster than the control. This is evident from the previous graph. Aeroponic lettuce on average was growing more each day than the control group. And as I mentioned before, the control graph demonstrated the height of some lettuce was decreasing. I concluded this shrinking was likely due to overwatering, and most of this conclusion comes from my experience with the first trial in overwatering, and also there was some error in my methodology, using only my finger rather than a device or something to measure the amount of moisture in the soil. So it's likely that based on the amount of water they were receiving early on, the shrinking is likely due to the overwatering. Finally, temperature was likely a confounding variable within my experiment, and while I used a heater to try to accommodate for some of the cold winter conditions, the low temperatures likely affected both groups, and this is evident from the graph as well. Both groups tend to dip at the same time as well as peak at the same time, indicating that temperature was likely affecting both groups, but the aeroponic group continued to remain above the control, proving that it was more effective. Finally, based on the measurement of water usage each day the control group was watered compared to the water usage of the aeroponic group, the aeroponic group proved to save water, also confirming my hypothesis. Now on to some of the limitations to my project. One of the first huge limitations was my sample size. Using only 18 lettuce could be one reason that for some of the inaccuracies of my data. My window of time for the project narrowed given the failures that occurred from the first trial as well, so the period of measurement also served as a limitation. Ideally, I would have preferred to measure out the mass of my lettuce, but given their small amount of growth and the limited time, this data was not collected. Finally, the yield of the lettuce was another piece of data I would have liked to compare, but given the time constraints, it was not possible for this project. Next, for future studies using my aeroponics system, these could involve different plants. While I chose lettuce, since it grows fast in the aeroponics system, other vegetables could be experimented with as well. Also, I believe adjusting the timer interval of the aeroponics system to be faster or slower could make for another interesting addition to this project, considering that the influence that adjusting my timer setting had on my first trial as opposed to my second trial was very significant. I believe temperature is necessary to measure in the future, especially because it was likely a confounding variable in my experiment and causing some of the dips and peaks in my graph. And finally, solar power could be another helpful addition to this experiment, especially because my focus is on sustainability, so it would be just another element to add to this project to make it even more sustainable. Lastly, I want to sincerely thank Mr. DeGroat for all of his help with my project and my research. Thank you to Mr. and Mrs. Nielsen for being such amazing chaperones on our Hawaii trip. Obviously, thank you, Mom and Dad, for all your support and help on the late nights in the freezing cold greenhouse. And finally, thank you to the Marine Chemistry class of 2022. I love you guys. So these are my sources. Are there any questions? Yeah. Okay, so the question was, did I use any supplements for the control as well as the hydroponic group? And that's a great question. Actually, the amount of nutrients that was in the hydroponic mix, which I had in my reservoir, lined up perfectly with the amount of nutrients located in the starting soil mix for my control group and also the potting soil mix. So the aeroponic group didn't have some sort of advantage when it came to nutrients. It was just lining up, the same as the control. Any other questions? Yeah.
How easy was it to set up the aeroponic system, and do you think use more traditional methods of gardening should switch over?
Okay, so the question was, how easy was it to set up the aeroponic system, and should people switch over to this method rather than traditional gardening? I would say that it was actually very simple to build. It took me one day. It wasn't that long. It took me maybe a few hours. And I had the help of some of my parents, so that made it easier. And I would say definitely it would be a good idea to switch. In fact, some gardening commercial manufacturers actually are advertising this product because, like I mentioned earlier, studies have been proving that it results in higher yields and it's just as effective, but it's also saving you more water. So yes, I would highly suggest more people to switch to an aeroponic system. Any other questions? Okay.
We're going to go ahead and take a...
Oh, wait, there's one more question.
Sorry.
Yes? The question is it true that it's my birthday today? And the answer is yes.
Yay, happy birthday. Thanks for sharing it with us. Wish I had known that. Hey, we're going to take a short break. However, we have a photographer here from, I don't know if they're yearbook or if they're the newspaper. Is that person here someplace? You would like to take a picture of us. I don't know what us mean, if that means the Marine Chem group or everybody here, or so why don't you come on over here, and then I know at least the seniors are going to get a picture. But come on over. Yeah, that's it.
of a self-sustaining pond and waterfall. I'm going to preface this first. My project's a little bit different than everyone else's in the fact that it's not more of a scientific project, it's more of an environmental design. And I mean by that, I'm not going out and collecting data and analyzing it, rather than solving a problem like caused by like environment change. So, a little background. We all know this, climate's changing, but Puget Sound is especially changing. Our watersheds are warming, facing rising sea, heavier downpours, larger and more frequent floods, more sediment in its rivers, less snow, and hotter and drier summers. Hotter and drier summers is where I'm focusing my project. Ephemeral or seasonal intermittent stream durations are shortening. According to the environmental research letters, intermittent streams make up half of Earth's waterways, and some are drying up 100 days longer per year than in the 1980s. This is no bueno. This is not good. This loss of water is having a catastrophic effect on urban wildlife. One of the animals suffering most from this loss is birds. Birds need this water to rehydrate, bathe, and cool down during summer heat months, or heat waves, but they can't do this if there's no water. But why am I doing this project? Because this project personally affects me. I live on a 2.5 acre farm, and this stream right here that runs down the center of it is actually an intermittent stream during the rainy seasons, running perfectly. But unfortunately, in the summertime, this is what the stream looks like, completely dried up. I have a bunch of farm animals, and as you can see, there will be no swimming or drinking going on in this stream during the summer, and we're going to fix that. So the question is, can we create a self-sustaining pond to supplement water loss due to the seasonal drying up of the local intermittent stream on my family farm? The first step is creating a design for this. And the design is going to be self-sustaining too, because we don't want to do any more damage to this environment. So we need a place to collect the water, which is going to be our roof. It's our watershed. Right underneath it, we're going to have a reservoir to collect the water. It's going to have downspout and gutters connecting them. Then a PVC pipe is going to run to our pond and waterfall location. We're going to have a waterfall to circulate our water, but we need power for this, so we're going to have solar panels and an electrical system for our waterfall. And then cables are going to be run from our electrical system to our pond and waterfall. All right, the first step, our watershed and reservoir. So our watershed is going to be our roof. It's 35 feet by 15.5 feet, or about 465 feet squared. And our water reservoir is 7 feet in diameter. This is important to know the dimensions because we're going to need to know how much water we're going to collect and how much water we can hold. So this is called a water budget. We're going to determine our water budget. A water budget is our precipitation minus our evaporation, so our rain minus how much water is going to be lost. The column on the far left is our precipitation unit rates per month, which can be found online. Washington State collects that, thankfully. We're going to multiply it, which is this column. We're going to multiply it by the area of our watershed, our roof, to get the cubic volume. Then we're going to convert into gallons. And then we're going to add the total the total gallons up, which is done in this column right here. And we're going to see that we have 8,878.9 total gallons, a lot of water. But we're not done with our water budget. We have to find out the unit rate of evaporation, but we're not doing that from our watershed this time. Instead, we're doing it from our pond because that's what's going to be evaporating. Our pond is obviously much smaller than our watershed, so our number is going to be much tinier. Again, it's the same process. We find the unit rate per month. We multiply it by the area, which is this column right here. Then we're going to convert that into gallons. And then in this far right column, we add them up to see we have a small 341.09 total gallons evaporating, meaning this is plausible. All right, the next piece of this is the reservoir. So the reservoir is a recycled 1,000 gallon fish tank. And then you can see right here in front of it, this pipe right here in front of the ladder is a part of our downspout and gutter system that leads from our watershed to our reservoir here. This is the connection pipes from the reservoir to the pond. This little blue thing right here is our flow valve, and essentially what it does is it restricts the water flow from our reservoir to our pond. And then this little faucet, it's kind of hard to see, this little silver thing is a faucet, and that's there to drain the water if it ever gets contaminated or there's any cracks or any problems or just honestly any reason at all we need to drain the water. That's there for. So this is PVC pipe that gravity feeds into our pond. The last part of our reservoir is the water overflow drain. This is super simple. As the water rises, the water is going to come fill up and then go out here, and it's going to be directed. Unfortunately, it doesn't really direct into anything except away from our reservoir and where people walk. So, yeah, it just gets the water out of the way instead of allowing it to overflow everywhere. All right, we're nearing the end of our water system. This is the construction of the pond itself. So right here, a 8-foot hole by 2 feet deep was dug by me and my grandfather. And then there was an identical fish tank that was 1,000 gallons also as a reservoir. And it was cut down to about 18 inches in height and just plopped in there and fit perfectly. Then a tarp was laid down. Right here, this tarp was laid down and then spray painted to figure out the design of our waterfall and what it was going to look like. We wanted to incorporate little pools for songbirds so that they could use this too. And then 1- and 2-man rocks were found on OfferUp for free, cheaper, and placed by me and my friends and my family. This is the float control valve. Its job is to stop the water from emptying out completely from our water reservoir into our pond. So essentially, it's a little pocket of air, and as it rises, it blocks off any more water from coming out of our pipe right there. And then if an animal goes in and drinks or evaporates, it's going to lower and allow more water to come up, filling our pond back up. That was the final part of our water system, and we're on to our electrical system. So this is a 3,600 gallon per hour bilge pump. It's typically used on things like boats, but we're using it for our pond because it creates a good flow. And now in order to power this, we're going to need our electrical system. So these are the mounts for the electrical system, and then this is the panels that were mounted to these mounts right here. The panels are two 270-watt solar panels connected in parallel to provide 12 to 13 volts, which are going to produce a total of 18 amps of current. The panels are mounted on the deck facing southeast direction to capture maximum sunlight. This is a solar charge controller. Its job is to control the flow of the current from our solar panels into our batteries. So it prevents, if there's ever a solar surge or a solar storm, which we cannot control, this will stop the siege of energy coming in and ruining our batteries and stopping them from exploding. This is, like we saw in the last photo, the solar charge controller, and me wiring it to our batteries. The batteries are two deep cell 6-volt batteries, which are wired in series to provide which are wired in series to produce one 12-volt battery. The 6-volt batteries are deep cell, providing 100 amp hours. Deep cell batteries are just batteries that last a long time. They're going to be found in things like golf carts. They're not really your car battery type battery. This is a really important piece for our electrical system. This is the timer. It decides when and for how long our system, our pump and waterfall is going to be running. It's an AVL 150 timer, and it is very difficult to program. This is finally last part of our project, and this is the spillway. It creates a flow. So instead of just having a hose at the top spraying all this water out, it's going to spread the water out and create a nice flow. And actually, if you look at the bottom here, you can kind of vaguely see the little pools that were created for our songbirds. Results. The watershed will provide 2,600 gallons of water to fill the reservoir by the end of November. This reservoir only holds 1,000 gallons of water, so the watershed is more than capable of providing the necessary water. Due to the long storage time, to keep the water free from bacteria, a mild solution of chlorine will need to be added. The reservoir will be covered to prevent evaporation during the dry season. And according to the Department of Ecology, they have approved rainwater from composition roofs for domestic use, gardening, and farms, meaning it's safe for our animals. electricity results. The deep cell batteries will provide approximately 100 amps per hour. The waterfall pump operates Monday, Wednesday, and Friday from 3 to 5 p.m. The waterfall pump requires 3 amps per hour to operate for a total of 6 amps used. Final results. The pond is going to evaporate 340 gallons per summer, and the reservoir will have 1,000 gallons with 660 in reserve. Water purification is maintained by circulating the water through the waterfall and adding Aeroponics pond clear chemicals. What does this all mean? Well, it means the development of the pond and waterfall system is going to adequately provide the much-needed water for the domestic and wildlife inhabiting the farm during the summer dry seasons. So in the end, it looked like this. And this is Connor and Mildred, and they loved it. Wait, I'm not done yet. Got ya. All right. So, some casing. Some future work could happen. Our reservoir needs some casing for it to keep debris out. Some fine-tuning needs to happen, some adjustments with our timer. And with our 7,000 extra gallons of water we're going to have, there could definitely be some use for that water, like irrigating some crops that the farm also has. And this is kind of my last big point as why did I do all this? I did it for the animals. I was on the internet and I saw this quote and I really liked it, so I put it in here. It says, it's not whether animals will survive, it's whether man has the will to save them. Since 1970, the population of birds in North America has decreased by 3 billion. That's not good, and it's because of us. We've taken their water sources, we've taken their homes, and we've taken their food. And you don't have to create a giant project like this to give back, but all these things cost less than $20 on Amazon. So I think if we started doing our part, maybe we could actually do some good. All right. And I want to thank Mr. Nelson, my grandpa, for just creating this program, for me able to do this, and for being my mentor and just really pushing me through, and my grandma for also always being supportive, and my brother, he was just always there helping me cut boards and stuff. Mr. DeGroat, you were always up at 8 o'clock with us too. That was a grind. My parents for always being supportive. Luke for helping me do some construction. My friend Nick for also helping me with construction. And Carlton, you helped me take drone photos, so that was also very useful for this. All right, and I'm done now. All right, does anyone have any questions? All right, Aiden? All right, so that's actually that. So the question was what was the total cost? And that's a difficult question because a lot of the stuff was reused. For me, even the solar panels were reused. I'm trying to think of the stuff we actually had to buy. Like the PVC pipe was reused, the fish tanks was reused, the roof was free. The batteries were probably the most expensive along with the pump. The pump was about $200, and the batteries together were about $200. So I say in total it was about $400 for us for this. But like I said, you don't have to go all out like that. You can Amazon $20. Do your part. Any other questions? Oh. Yeah, so it's placed in the middle of the stream, so that way in the winter months it can collect water. But during the summer months it can kick in, and so when the stream completely dries up, it's able to just function on its own because it's been holding water during these rainy seasons, if that makes sense. Why didn't we have the waterfall running every day? All right, that was the question. To not drain the batteries. The strain on the batteries would be great, and the waterfall is mainly run to just circulate the water. The water doesn't really get stagnant in between that period of times. It stays like well used, so that's why. Oh wait, yeah. Yeah. So, like, you have chemicals in all of our water. All right. The question was chemicals with the water. So I think she's asking about you're asking about the chlorine in the reservoir and stuff. Yeah. So there's actually a set standard of amount that there's a little table given to. I'm trying to it's like a standard, like a regulation. For like farm animals and stuff, so you just follow those pretty carefully. All right. Thanks, guys.
Ryan. Oh my God. Working? Wait. Hello? Oh, yeah. Okay. Oh, thank you about that. All right.
Hello, my name is Phytophthora Hegstead,
And I'm Carlton Degwa.
and our project was on the utilization of drone photogrammetry to observe eelgrass coverage within Titlow Marine Preserve. Eelgrass, aka Zosterae marina, is a species of seaweed that's commonly found along the world's oceans, including Tacoma's local marine preserve, Titlow Beach. In the eelgrass's abundance, it acts as a major factor for life in the ecosystems it's found in, making it a foundation species that works as food, shelter, or nurseries for other local sea life. They also act as important nutrient recyclers and carbon sinks. Unfortunately, the presence of a slime mold known as eelgrass wasting disease, aka Labyrinthula zosterae, has been known to kill fields of eelgrass due to the discoloration it brings to those it infects, making photosynthesis difficult to the infected. In order to detect L. zosterae in its early stages, researchers have recently begun to utilize drone technology and GIS softwares in order to quickly identify an infected area and prevent further spread of the infection. However, as far as we know, there's not been a noticeable local spread of Labyrinthula at Titlow in recent years.
Before we introduce our project any further, we first want to define some key terms within our project to help the audience understand our procedure and process. The first of these terms is photogrammetry. Photogrammetry is the science of obtaining information about an environment or physical objects through recording, measuring, and interpreting photographic images. Though there's a variety of ways photogrammetry can be utilized, its most common usage is within cartography, where cartographers will use aerial photography from drones or UAVs to map large-scale areas and buildings. You can think of photogrammetry as a jigsaw puzzle, in which each image taken is a piece of the puzzle that, when compiled through a geographic information system, creates a cohesive image. The second term we'd like to define is a geographic information system. These are softwares that analyze photos taken by UAVs and drones and convert them into raster or vector maps, which can then be viewed and visualized in 2D or 3D. However, for these softwares to be as accurate as possible, a few rules need to be followed when collecting data. For instance, when analyzing a 2D landscape such as a plain or agricultural field, the images must be taken by the drone at various altitudes at a constant overhead angle in order to capture levels of clarity and to give the region a realistic depth of field as much as possible. This allows the software to render specific regions of the given area as realistically and clearly as possible instead of having to guess how certain parts of a landscape look like when magnified. For the same reason, when taking aerial photos, there must be at least a 20 to 40% lateral overlap between the top and bottom of the images and a preferred 60% to 75% forward overlap between the left and right sides of each image to provide the software with enough information to create a fully rendered map instead of leaving gaps that could alter the size, shape, and cohesiveness of the map. The software utilized within this study is ArcGIS's Drone to Map, one that has been specifically used in the past in similar studies for mapping eelgrass populations
within estuaries and marine preserves along the west and east coasts.
The overall purpose of our study is to determine if ArcGIS is a viable tool in determining the coverage of eelgrass population within Titlow Beach. To do so, we utilize drone photogrammetry and the drone-to-map geographic information system to create accurate maps of the studied area to track changes of eelgrass within the marine preserve. These methods set drone position, time of day, tide level, image overlap, and image amount as the independent variables of the study. The dependent variables are the map's accuracy, clarity, and detail. These maps act as key of determining what changes occur over time. Our hypothesis for the study is that ArcGIS will be effective at mapping Titlow's eelgrass population and provide maps that prove as such. We believe this because our study follows a similar approach that other researchers across the west and east coast have taken in similar studies. However, within recent years, there have been little to no report on critical environmental factors in Titlow Marine Preserve that would show in disparity in eelgrass population. Factors that were taken into account while conducting research fell into two categories, one of factors beyond our control and the other of those within. The uncontrollable factors included weather conditions, tide conditions, water clarity / angle of the sunlight, and wildlife interference. If any of these factors denied us from collecting our data properly, we likely would have had to wait until another day to continue the data collection. Tide level and water clarity were specifically prioritized at times because we had to adjust our schedule around what time of day allowed the water to be most visible and tides to be far back enough to see further eelgrass beds. The factors within our control consisted of drone altitude, number of photos taken, image overlap, and camera angle. As expected, we took full command of these factors, making sure to prioritize the veracity of our data collection. Of all these factors, we attempted our best to ensure that our data was not obstructed and completed to an optimum level of accuracy.
Mask this one. Determining the requirements need to accurately take aerial photogrammetric imagery. We narrowed down our list of drone options to the DJI Mini 2 drone. This drone was cost-effective, met our criteria for high-quality photography, and has a built-in global positioning system, geotagging each image taken with the latitude, longitude, and elevation of the drone at the time each of the aerial photos was taken. Due to the weight of our drone being 1 gram less than the required recreational drone FAA course requirements, we were not required to undergo any proper training or follow any specific regulations when it came to conducting our research. After acquiring the DJI Mini 2 drone, flight dates were reviewed in order to gain permissions from Tacoma Metro Parks, taking into account the optimal weather and tide conditions for drone flight. These dates were 2 weeks apart within the span of 2 months, and thus data collection also followed the same pattern. The photos taken over the following months of the summer were taken through manual flight, flying the drone over the study area, and taking pictures via controller. Here, the camera angle was set to either 45 degrees or 90 degrees, and the photos were taken at altitudes of 50, 100, or 200 feet. After the collection of all aerial photography, the ArcGIS software was used to convert the photo sets into 2D maps of Titlow Beach, able to be reviewed and compared to one another. A designated sample region for each map was analyzed using the aerial tool within the drone-to-map software. Each sample region was calculated by hand, plotting points around the boundaries of the given region. We then used the area of the shaded region to determine the area of eelgrass based on the removal of negative space such as sand, boats, and anything on the map that was not green in color or considered eelgrass. Sample proportions of eelgrass coverage to the total area was calculated for each map, with 3 sets of null hypothesis tests created, one for each sample proportion. Each hypothesis was tested to see if any of these proportions would be consistent with all 3 maps. As you can see up here, these are all 3 of our tests we made. This is for the first day map, this is for the second day maps, and this is for the second day maps right here. This is for the thirds, this is for the firsts. And the red colors indicates where the hypothesis was rejected, and green means it was failed to reject.
For our maps we have, on the left, the rendered map unobstructed from view, and on the right, the same map with the total area analyzed and measured. Thank you. As you can see, the map has line segments at certain points. This is due to our drone's camera angle being set to 45 degrees, creating an inconsistent top view of Titlow's to create our map with. Near the end of day 1 data collection, the camera angle was fixed to 90 degrees and continued the flight at 100 feet altitude. Tide level and sunlight exposure during the day were optimal to see eelgrass far past the shore, allowing for high visibility of eelgrass beds. From our analysis results day 1 data, we were able to determine that the eelgrass covered an area of approximately 14,757.0289 square meters out of the total area measured of 15,447.9529 square meters. This means that 95.53% of the total area measured was covered in eelgrass. Our maps for day 2 are presented in the same order as the previous slide. This day has slightly less light and worse angle of sunlight exposure, but made up for it by having the lowest tide out of all 3 days, allowing us to go further past the shore to capture dense populations of eelgrass beds. The flight path and compiling of images made the map less complete with jagged borders, but did not affect eelgrass visibility. The camera angle remained consistent at 90 degrees, and elevations ranged from 50 to 100 feet. Near the end of data collection on this day, a few photos were taken at 200 feet to attempt to fill in gaps in the flight path and add more cohesiveness to the rendered maps. For day 2, the total area was calculated at 10,900.7839 square meters, while the area of eelgrass coverage was at 10,209.8599 square meters, meaning the eelgrass covered 93.67% of the total area measured. The day 3 maps are presented again in the same fashion as the past 2 slides. During data collection, the camera angle remained at 90 degrees at all times, while elevation ranged to 50 to 200 feet, with tide levels lower than day 1's and approaching but not surpassing the depth of day 2. Sunlight exposure was the lowest of all 3 days due to partly cloudy weather, though sunlight could not be used to our advantage in terms of low visibility as much as day 1 and 2. Day 3 was our most complete render out of all 3 maps, with no visible deviations in color or shape. A uniform border and a remaining within the bounds of its flight path and all images taken. With the middle map giving the total area measured 14,971.9669 square meters, the eelgrass covered an area here was also made up an area of 14,281.0429 square meters, making the eelgrass cover 95.39% of the total area measured.
Our results deemed our hypothesis as inconclusive in determining the efficacy of ArcGIS as an effective tool of determining eelgrass coverage in Titlow Marine Preserve. Since there was no outcome where all 3 maps failed to reject the null hypothesis of all 3 sets of hypothesis tests, our maps data cannot prove that ArcGIS is an effective method of eelgrass coverage analysis, but due to our long list of limitations affecting our procedure of research conduction, we cannot fully come to a conclusion on the efficacy of the ArcGIS software. In other words, everything we did was a waste of time, and all of our maps had no, we'll explain that on the next slide actually. So as you can see from our graphs, I'm gonna go off script for a bit. As you can see from our maps, our graphs, there's a disparity in eelgrass coverage for all 3 days. That little dip there was pretty drastic. So the left graph here is our map, it shows each value we got for our hypothesis tests. It says 5,000 there. The numbers app is pretty stupid, and so you can't put decimal points. So I put 5,000 to just show the drastic, because we did 4 sig figs, so I just put them as 1,000. But the values, the graph is still accurate regardless. So basically, if at least one of the hypothesis tests had shown that all 3 maps had failed to reject the hypothesis, then at least all 3 points would be at the same point. But because of this day right here, that does not happen. As you can see, this is day 2's trajectory, and this is days 1 and 3. And it should follow days 1 and 3's trajectory, or at least they should be on the same points at all times, but that didn't happen. There was a lot of reasons for that, primarily manual flight, because the area we went to we did manual flight, which you shouldn't do when mapping drones because human error for one, and automated flights normally have the same path all the way through, so you should have seen the same map for all the 3 days. Clearly, you did not see the same maps for all 3 days. As you can see here, again, those were our flight paths and our images that we used for those days. Clearly, there was something wrong. So this was our first day up here. As you can see, the flight path drifts off drastically. The map should have at least covered around this area over here, but due to our lack of experience. And also the area there is next to Tacoma Narrows Airport, and technically you shouldn't fly drones there, but we, don't tell them that.
For science.
What was that? Oh yeah, we did. We used the software and it said you couldn't fly there, so we did it by hand, and clearly that was a mistake, but we moved. So our biggest limitation, as I just mentioned, was the human error as a result of our manual flight. Normally we suggested that we use automated systems, but we didn't do that, so because of that, our resultant maps varied in size and shape and also consistency. As in day one, there was a drastic difference in terms of the rows of images that you saw because we did 45 degrees out that day, and because of that, it showed the images that were in front of the drone instead of beneath the drone, and that's why there was varying rows instead of a cohesive image. Surprisingly, day 3's map was actually pretty decent, even though we still did it the wrong way every time. I don't know how that happened, but God was on our side that day or something. I don't know. Oh, the birds were a big thing. Birds were surrounding the drone constantly because the noise, it was white, it was like 200 feet in the air, so obviously it's going to attract a lot of noise. They loved our drone. So we had to take it back and break the flight path multiple times, so we didn't know exactly where the flight path was, because as you can see, it shows up there, but it does not show up on the drone software. They had to bring it back every time there were birds disrupting it, which was pretty time consuming and also broke the pattern multiple times as well, which is why it's very hectic, as you can see. Analysis by hand. Okay, that software, we know it's, with past studies, it definitely shows that it's an effective method, but whoever designed that area mapping software should not be a programmer. That tool was horrendous to use. And Fi was next to me, and I was trying to keep my cool, but I swear I was gonna break that monitor because it was so frustrating. That was also a pretty distinct method because we couldn't map it accurately because you can't move the map when you're plotting points for the area. So we have to zoom out to see the entire Earth, zoom out, and zoom all the way back into that specific point, plot the points, zoom out again. And I'm kind of wasting time, but just know it was hectic. So future classes, don't do that. Don't do that ever.
Which brings us to future research.
Yeah, the future research. Yeah, there you go.
We are hopeful that future research conducted on local eelgrass coverage in Titlow Beach ecosystem will continue. Our hopes is that future generations of marine chem students can take our research and expand on it in a variety of ways, maybe the right way. First of which would be to use automated systems over manual drone flight, as we explained before. We would also suggest using different types of drones and maybe even cameras with different spectral imaging, such as ultraviolet, infrared, and LiDAR. The comparison of ArcGIS to other geographic information systems, as well as comparing like the different autonomous flight systems. And also potentially studying Labyrinth through Zostere itself and looking more into a more hands-on way of identifying it and tracking spread, preventing it from further harming eelgrass ecosystems. And near the end of our study, we were suggested the idea of true sourcing to add as another reference to expand on the accuracy of our maps. However, we were too far into our project to apply this method. We would recommend comparing this to automated drone photogrammetry and determine any differences in effectiveness. And finally, we hope to see future generations of marine chem students continue to monitor Titlow Beach in a similar drone flying GIS using manner. The continuous use of drone photogrammetry and GIS systems has tons of potential uses for future studies, and we just wanted to be our first to kind of put our foot in the door. I'll say thank you.
I'd like to say thank you to everyone that has aided us in completing this project. First of all, thank you to our families for putting up with us for 18 years of our lives and for especially supporting us during this last trek through senior year and working on this project. It was we love you all. So, let me do that. So thank you to our parents, and then thank you Mr. DeGrow for guiding us on the right track towards this project. I'm gonna let you know this project had like multiple different titles and multiple different purposes throughout the year, so he helped us narrow it down to this one. And it was very helpful, and he helped us figure out what we wanted to really focus on and revolve our project around. So thank you, Mr. DeGrow, and for being a great teacher and undisputably the best van driver, no debate at all. So I don't want to hear any complaints of any of you. Thank you, Mr. and Mrs. Nielsen, for the amazing Hawaii trip. It was a great time to be with the entire class and just have so much fun exploring Waikiki, all the homies. Oh yeah, I got slumped a lot, I'm not gonna lie, as you can see up there. And thank you again to the Marine Chem class of 2022. Y'all are the best. I'm so glad I can call each and every one of you one of my friends, so thank you.
Here's our sources. And any questions?
Oh.
Grace.
Yeah, Grace.
So how did you determine what was eelgrass and what was just like
Okay. The question was, how do we determine what was eelgrass and what was kelp or algae? Very good question. We actually didn't do that. So basically, if we saw that it was green and looked like grass, it was eelgrass. So that's what we did. So yeah. Any other questions? Yep. The question was, were we unable to use Aeroponics software because it was too close to the airport? And the answer is yes. Basically, if you see the software, it will show you a map of areas you can and can't like map, and it'll show you different elevations you can use. The short was the cutoff for where you can fly the drone, so we can't use it like almost any other automated software. So that's why we can't use it at all. So we have to do it by hand. Clearly, poor choice, but we do what we do. Any other questions? Oh, yeah, Salve in the back. This one?
I would suggest studying over the same area of Titlow Beach. Only difference is that when you try to use an automated system, try to find one that doesn't have as much limitations, or if needed, get the special permissions needed that would allow you to use certain systems in the area.
Do I need to turn this off?
All right. I know many of you are at your wits' end, so I'll try to keep this short and sweet. Let's talk about some avalanches. During the 2020-2021 winter season, 37 outdoor recreators died in avalanches in the United States. 26 of these skiers, snowboarders, and snowshoers died in the month of February alone due to an incredibly weak snowpack. These death tolls surpassed deaths from both earthquakes and commercial airplane accidents in the last 20 years. Avalanches are truly one of nature's killers. Advances in technology and closures of ski resorts due to COVID has led to a backcountry boom. Backcountry equipment sales are up 150%. This recently has led to an increase in death tolls, which is expected to continue to climb. This middle photo is of the Wilson's Glade avalanche in Utah last February. And you can see that if you're lucky enough to avoid being strewn through trees, you're more than likely going to be killed by snow immersion. And finally, poor avalanche forecasting causes the closure of major mountain passes, such as Snoqualmie Pass, leading to millions of dollars of economic losses. All avalanches involve one simple physics problem, where the force of gravity overcomes the force of friction, holding the snowpack together. Slab avalanches are the predominant killer of these winter recreators. They involve four things. First, you need a slab of snow, a slope 30 degrees or greater, a relatively cohesive slab so that the snow can propagate a fracture, a weak layer, and a trigger. In this case, on this photo, a skier, rider, or hiker, but often can include wind, rapid loading of the snowpack, and occasionally even earthquakes. There are many different types of weak layers in the snowpack. This includes depth hoar, near-surface facets, grapple, but pictured here is surface hoar. But for simplicity's sake, weak layers are generally just marketed differences in density in the snowpack. Thankfully, however, we can dig snow pits to look for these weak layers. And in these pits, we can form a variety of different snowpack tests, most commonly being the extended column test, which I will show you here. Hopefully this works. So this is me performing the test. And here we're isolating the column of snow. It's 30 centimeters by 90 centimeters. So I'm isolating the column. And right now so basically you're trying to create an area where you can create a mini avalanche. You isolate the column so you don't need much force. Here's me just continue to isolate.
So we're looking, because we've done the test a couple of times, we're looking at these two different layers in the snow, one here, and then another one here. It might be kind of hard to see in the camera. But as we're doing the test, we'll be watching to see if that layer breaks, propagates, fractures, whatever. So I'm starting with the wrist tap.
So I'm showing you the extended column test, and it starts with these wrist taps. You're applying force on this isolated column of snow.
I saw no signs of fracture.
And that was after 10 taps.
The elbow tap.
That's elbow tap.
And I saw it fracture slightly.
So the snowpack failed, and it just fell out.
Collapse, but no propagation across the snowpack. You can see that this block is still locked into the snow.
So there you may have picked it up or not, but you just witnessed the problem that I saw initially learning about this sort of thing. The force on the column is entirely determined by a human conductor, and therefore it's subjected to lots of error. So we're now going to look at some videos, some slowed down, of a professional performing the same exact test, using varying levels of technique, and somehow assuming to get the exact same result. So you can see totally different technique, totally different force profiles, trying somehow to get the exact same result of whether or not the snow is safe or not to recreate on. So my objective is to modify the extended column test in order to decrease variability and error. And to do that, well, we first needed to figure out, well, how much variability, how much error are in these different taps. And to that, I was fortunate enough to get a force plate from the marine chemistry lab and got some data on how much variation there is, and really just kind of revealing the core problem what's going on here. So right here is on the y-axis we have newtons of force and then our different trials, and this is all of my force from the elbow of the extended column test. And you can just see how much variation there is in force on these different tests. I remember one test, I believe it went from 105 newtons to on the next test it was over 150. So it increases of like 50%. So we then averaged our data from our 3 force stations and got the values we needed to emulate in our new test. So we essentially entered a period of brainstorming on this new instrument. And the objective was clear, but it had to be convenient to bring in on a long hike and it had to prove its worthiness and its efficacy. After consulting with a family friend who worked at Blue Origin, actually, as an engineer, we surmised some sort of spring-loaded system. However, after drafting the device, it became obvious that it would be over-engineered and overly complex and wouldn't really satisfy our criteria. So after some thinking, I ran across this random government document from 1996 about the stuff block test, which involved a 4.5 kilogram block rested on an isolated column and then raised and dropped in 10 centimeter increments until failure. The article reads, as a loose guideline, drop heights of about 0 to 200 millimeters indicate mostly unstable snow conditions, 300 to 400 millimeters indicate moderately stable, and so on. So turns out the snow block test was fairly popular a couple of decades ago, but it was replaced by the ECT because of its convenience. A little interesting factoid that solidifies the lack of science in this field is just completely arbitrary force values that the stuff block is dropped from. So my goal was, well, let's make it better. And this is where things get a little bit gray in terms of trying to emulate the force values from the force plate into a new test. So we first have our physics equation that is just the net force is equal to the change in momentum over the change of time. As we move down our deriving equation, our momentum is equal to mass times velocity. Velocity is equal to the square root of 2 times gravity times height. And finally, we get this final equation of here at the bottom. And well, we need to find our mass, we need to find our height, we have gravity, and we need to have time. So how do we find time when we don't have mass or height? That's very difficult to do. You can't do it when you have that many variables. So I just kind of dropped what I'd think, would make this amount of weight, what I'd make this amount of force. And you can see in this video, it'll play. This is that amount of time, force of impact for the slow we plugged into our equation. And a little side note, other than the limitation of our study being finding this time, was different snow surfaces produce different impact times. Different impact times affect the net force of the equation. So moving on, we have our equation here. And this is our wrist tap, the average force of our wrist tap. This is the average force of the elbow tap. And the time changes from one to the other because obviously it's more force. And basically, I was looking on this graph for a most convenient spot to put my height and my mass. And that ended up being, we got these two values from the wrist and shoulder. 0.03 seconds from 3 centimeters and 0.06 seconds from 30 centimeters, meaning 3 kilograms will be dropped from these heights to emulate our net force. As you may notice, we got very lucky with this, and that was most likely pretty accurate. So what I'm saying is we got pretty lucky with trying to create the same amount of time because, well, when I was trying to find time, it was essentially 3 kilograms from 30 centimeters, which was just completely random, but we got it right. So we performed this test, and I'll show you what the test looks like.
This is 3 kilograms. So with the hanging fish scale kind of thing. And this here, and I can see where it is, and drop it. 1, 2, 3.
So this is just sped up. So we performed this test, the Leo block test, I named it.
125 centimeters and 30 centimeters.
So you can see this is me just basically simulating what the elbow taps were, and I saw the collapse.
You can see this line failed right here, and this goes smooth out. So we got ECT.
So we essentially got the exact same result. And data was collected, so we performed this test, the Leo block test, 30 centimeters away from the extended column test in the same pit. So we dug 15 total pits in total. Each pit had 2 tests in it for a total of 30. We dug the 15 pits. The first 5 were on a solar aspect, the next 5 were on a shaded aspect, and finally the last 5 were on a polar aspect, so facing north. And here are our results. So after these pits, this is the data we found, essentially all over the place. On the y-axis, you can see how many taps the block took to fracture, and the x-axis are different pits. So the ECT is in blue, and the LBT, the Leo block test, is in red. Yeah, and to clarify, after 10 taps of the wrist, the conductor moves on to the elbow. So from the data, it seems like the first pit had less spatial variability in the snowpack, meaning the snow was more homogeneous. This was a slope facing the sun, which often leads to solar warming and rounding of the snow crystals, which homogenizes the snowpack. And as for the remaining sites, things were a little bit all over the place, but both tests trended towards higher stability results in the third site, which demonstrates that both tests find similar results. So, in the end, what did we see? Well, no observable decrease in accuracy with a normal ECT, and anything that could be deduced as observable would be difficult to prove significant due to spatial variability in the snowpack, meaning whether it be down trees that create different temperature gradients in the snowpack that change basically the structure, the makeup of the snow. And each test seemed to do fairly well at predicting avalanches, although not one better than the other. Each showed no real surprising result. And just for some interesting observations we had, the snowpack failed across the column when the secondary test was performed. What this means is basically after I did the initial test that you saw in the videos, I did a secondary test on what was remaining of the block, and that propagated across, that propagated a fracture, what was actually an unstable result, which is also an opportunity for future research. Something is going on where there's some non-observable fracture of the snowpack that's going on that we can't see. And this is very important. The Leo block test has also lots of room for human error and discrepancy due to drop height. Maybe you notice in the video, it's not exactly perfect me dropping it from these heights that are known. So someone could modify this test in order to standardize the drop heights, and that's an opportunity for some of you guys. And a modified version of my test could be useful when multiple professionals are attempting to track a trend. And what I mean by this is, well, for example, my data was collected in the Sawtooth Mountains of Idaho, and the Sawtooth Avalanche Center is supposed to predict avalanche danger for a area of 3,000 square miles for 3 people, and that's basically impossible. So if you were able to have a standardized test, they could give you a better view of what the avalanche danger is and the trending of avalanche, if it's trending towards stability or instability. Finally, here are my sources. You can see they're relatively short because, well, there's only 215 scholarly articles ever published on the topic of avalanches, and just 15 of them are on the extended column test. And this is kind of a theme that I was encountering throughout my research project. There's just not a lot of information, and there's very little real scientific information here. And just a little thing, you can see just names coming up again: Simon Hoyce, Birkeland. These are guys who are working at Montana State, and it's basically the only place that researches avalanches. And also half of the articles that I was reading were just meta-analyses of other research. Anyways, just kind of not well studied. And I want to give a huge shout out to my dad, who brought me on the trip that we got to collect the data, and for funding my passion and just being a great dad. To my mom for her endless love and support. Steve Walton, who was the engineer who initially helped me with the device. Mr. Chase for helping me with the physics problem. Tashi right here, looking forward to more great adventures. And the marine chemistry class of 2022. It's been a good 4 years. Thank you. Any questions? I was going pretty fast and a lot of information. All right, Grace. So this is an interesting question. The question is, how did I choose the location? And it's been a very wild winter season. As you may have noticed, it hasn't really rained significantly over the last month, and there was a slow start to the winter. So I had needed to find a place where there was some avalanche danger, but not a lot. The lack of snow made it difficult to do around here, and the maritime snowpack, meaning it's closer to the ocean. The snow is warmer here and more homogeneous, so I actually had to go somewhere in an intermountain climate in order to find snowpack instability. Any other questions? Joey, I know you want to ask a question. What's up? So the question is, how did I choose to use a weight of 3 kilograms? If I go back to my graph, on the x-axis we have, I believe this is, on the x-axis we have weight in kilograms, and on the y-axis we have height in meters. So I just found okay, what's the most convenient to use? And also, luckily, it matched with my time, and that was 3 kilograms. Thank you.
All right, I'm Aiden Moore, and this is my presentation on the effects of perceived sex of target on the self-disclosure of adolescent males. So first, I'd like to start off with some definitions and a little bit covering past research. So self-disclosure is defined as sharing about yourself with others, how much you share, how intimate your sharing is. So high self-disclosure would be sharing frequently and with a high level of intimacy, where low self-disclosure is sharing infrequently with a low level of intimacy. So this varies by sex. A lot of studies have shown this. Naturally, females tend to disclose more highly than males do. And so this leads to a bunch of differences when moderated by target sex. So males talking to males tend to have low self-disclosure. Females talking to females have high self-disclosure. But when you have these cross-sex pairs, females talking to males or males talking to females, it's sort of in the middle. So this is explained by a reciprocity effect, where by high self-disclosure by one partner leads to high self-disclosure by the other partner. So you sort of have people meeting in the middle when there's differences in self-disclosure. So in the middle there I talk about target sex, target self-disclosure. So target sex is the sex of the person you're talking to or the person you have a relationship with, and target self-disclosure is the self-disclosure of the person you're talking to. So given natural sex differences in self-disclosure, these variables end up being linked. So if you're talking to a female, your target sex is female, and the target self-disclosure is also, it's high. And if you're talking to a male, target sex is male obviously, and target self-disclosure is generally low. So because of this linkage of these variables, it's sort of really difficult to study them separately and sort of determine which is responsible for an effect. So a lot of studies have been unable to determine the difference between these, many pointing to target sex as the variable responsible for differences in self-disclosure. But because they're always studied together, it's sort of impossible to know which is more important. So which leads me to my purpose. So my purpose was to conduct experimental research on the effect of target sex alone on participant self-disclosure after controlling for the reciprocity effect. And so after controlling for this, I predict a slight difference because gender stereotypes are still present and people would expect that females would share more. So I studied adolescent males in this from public and private high schools in Tacoma and Gig Harbor. They were recruited through social media, and all of them completed a consent form. So my procedure, I want to talk really about how I controlled for this reciprocity effect and what I actually did. So I randomly assigned participants to a target sex condition. So they were either told that the partner they were interacting with was a male or was a female. In reality, the person they were talking to was a confederate, which means they were a part of the study pretending to be a participant. So I essentially catfished all of my participants. Yeah, so the reason I did this was to control for target self-disclosure. So because every partner was talking to the same person, who, as I discussed on here, who was giving the same answers to each question, target self-disclosure was kept constant throughout all the research. So by eliminating that difference in reciprocal effect, I could really see what target sex was doing on the differences in self-disclosure. So my procedure was online. This was done basically out of necessity because I needed one person to be able to pretend to be both female and male, which doesn't tend to work as well in real life as it does online. But I did find a number of studies confirming that the reciprocity effect is still present online, as well as talking about self-disclosure in online settings versus face-to-face settings. The results are inconclusive. So there are some studies that claim they're the same, some studies that claim people disclose more online or disclose less. So that part is it's unclear whether this is applicable to face-to-face communication, but it seems that it's likely that it is. So the procedure, my discussion questions, there were 10 of them, and it took about 45 minutes. My discussion questions were taken from an Arthur Aaron study on an experimental generation of interpersonal closeness. So these questions are sort of designed to get people close to each other, which is why I'll use them for this task, because people tend to disclose more if they have something, I guess, personal that they can actually disclose about. So both the Confederate and the participant took turns answering each question, with the Confederate answering each question the same for every participant. So after the discussion, they were given a questionnaire to determine a number of values. So as you can see, these were the items that they were given, and each one they responded to on a scale of 1 to 6, with 1 being they strongly agree with the statement, and 6 being they strongly agree with the statement. So the middle questions, 2 and 3, were chosen to assess disclosure given, so how honest they were and how vulnerable they were. And the fourth question was disclosure received, so how honest they felt their partner was. And the first question was sort of just asked to determine to what extent these questions induced a sort of feeling of familiarity. I used Google Forms and Google Sheets to collect and store my data. The data analysis was done through MATLAB, which was used for statistical analysis and visualization. And I used independent samples t-tests on all dependent variables across target sex groups. So I'll explain this graph with my handy-dandy laser pointer. So the M is mean and the SD is standard deviation. Each one of these rows are the question items, and these are the male responses and these are the female responses. So male means the gender they believed they were talking to, and female means, yeah, as opposed to the participant gender, it's their group gender. So some things to notice: all responses, these are averaged across 10 participants in each category. All are above 3, which means that every answer was at least slight agreement. And although all the female responses were on average higher than male responses, these t-values show that these differences are not significant, which is confirmed by all the confidence intervals containing 0. An eta squared is a measure of effect size, and so these effects range from very weak to weak right there. So not a lot of effect of the independent variable, which is the target sex, on the actual difference in self-disclosure. So some key findings. There is actually I found there was no significant difference based on target sex alone. And so this might seem to be a break from past research, given that a lot of people have found that target sex does actually have an impact. But because I sort of controlled for target self-disclosure, thereby removing any reciprocity effect, this study is really not comparable to past research. And so this sort of would suggest that that the difference experienced in past studies on target sex may actually be due to reciprocity instead, because these past studies are using both variables, so they have reciprocity and target sex, and it shows a difference. And there's also studies that show that just reciprocity alone, independent of sex, can cause differences in self-disclosure. So because of those two things, it leads me to conclude that differences based on target sex are not real. So target sex alone has no impact on self-disclosure. So some limitations: I had a small sample, only 20, which might seem large, but in this case it really was not as large as I would have liked. With a larger sample, I might have been able to detect some small effects that I might have experienced, which would have confirmed sort of what I expected, some small difference, but who knows? So a larger sample would have been nice to sort of confirm that. The researcher, who was me, was the Confederate. So because I knew the hypothesis and I was sort of the one conducting these conversations, it opened it up to a little bit of bias, which I tried to avoid, but sometimes it's unavoidable. Another limitation is that all responses were based on self-report. So someone could say that they were being really vulnerable or that they were really honest where they really weren't. And so I sort of have to take all these with a grain of salt, given that people could have just said whatever they want on my questionnaire. But I hope that they didn't, and in an ideal world, these results are perfectly accurate. So further research could sort of compare directly what I'm positing by comparing the target sex and target self-disclosure variables to find their relative importance, so which would, confirm or deny what I am just merely assuming, I guess, or guessing at. So in conclusion, my results may diminish the importance of target sex in favor of the variable target self-disclosure, because sort of as these things are progressing in time, and there seems to be a shift recently away from like tough guy masculinity culture, more towards more self-disclosure really. These variables of target sex and target self-disclosure might become less linked, and they might not be as the sex differences decrease, these reciprocal effect might not be as gender influenced. So you might have men that disclose really highly, which would sort of break the traditional model of target sex, because the idea that men disclose low would just not be true anymore, I guess. So as these sex differences decrease, you might see this variable of target self-disclosure becoming more important because it sort of explains more cases. I'd like to acknowledge my mentor, Dr. David Moore, who happens to be my dad. He's also a professor of psychology at the University of Puget Sound. He was really helpful in sort of helping me to write the paper, and throughout the review process where I had to get approval in order to do this because my research, contained human subjects. I'd like to thank my mom for being immensely, great source of support. Even though she wasn't actually my mentor, she's also a psychologist and was a great resource. I'd like to thank my participants because I couldn't have the data without them. And I'd like to thank Mr. DeGroat for being a great source of support, and the Marine Chem class of 2022. Yes.
Wait, what is the measure of self-disclosure? Like, can you give a sense? Does it mean you say a lot of words? Did you share something really deep?
Sure. So as I on my questionnaire, I had both honesty and vulnerability. So those sort of combined show a picture of what self-disclosure is. So it's not just you talk a lot or you're honest, but it's sort of a combination of all those factors. Where if you're honest and you're vulnerable, then you can sort of be said to have high self-disclosure, and sort of if you're not honest or you're not vulnerable, then you can be both low self-disclosure. But as my results showed, pretty much everybody in the study had a high level of self-disclosure, which would be uncommon, I guess, because all my subjects were male. In an ordinary case, you would expect that they would disclose low because that's sort of been the past research, but they all disclose very highly, which I would assume is because of that reciprocity effect. And the answers that I wrote for the Confederate to give were all high self-disclosing, and so every participant sort of was moving towards that high self-disclosure of the Confederate, which sort of explains why all my values were super high. Jalen. Yeah, so the question was, what were some of the questions I asked the participants? I had a PDF of 20 questions that I sort of dropped into each conversation, and they started out sort of easy. The first question was if you could have dinner with any person, who would you choose and why? Then it progressed into like sort of what's like an embarrassing moment in your life, like how happy was your childhood, and then sort of ended up with sort of more like really personal questions about like which family member's death would disturb you most. So yeah, it got pretty personal by the end. Yeah. Grace. The question was, were all the participants the same age or grade level? And the answer is no. although they tended to be juniors and seniors. I think I had all juniors and seniors and one sophomore. I did not account for age, mainly because I forgot to ask age until the end. So that was sort of my bad, and if I were to do it again, I would ask age. Joey. Yeah. Joey seems to be hinting at a sort of an acknowledgement I may have neglected to make. I would like to give a special thank you to Joey Vaughn, as he was the only participant whose research session I slept through, because I was a fool and I scheduled him at like 8 a.m. on the day after Halloween. So that's really on me for scheduling him then, and I apologize, Joey. Thank you. So my interest, I mentioned the Arthur Aaron study. It's something that my parents as psychologists had told me about for a while, and that was sort of the study that was in pop culture as like the 30 questions that lead to love, because in an initial research thing, they'd had 2 subjects that went on to get married, and sort of this study became super popular, and so from that I sort of was interested in closeness, and it sort of just progressed and developed into this project. Yeah. Want the mic? If I can get it out.
Right. Hey, so this is the part of the show where if anyone would like to present anything they've been studying, come on up and we'll have a question and answer session. Is there? No. Well, if there are no other research presentations, we're going to call it a night then. Thank you for coming this evening. Our next steps with these guys, we're going to find out Thursday who gets to present orally at the Washington Junior Science and Humanities Symposium. And then all these guys are going to be entering into, it's now called the Discovery Science Fair, what's out at PLU. And that's happening in March. And our juniors are underway right now, kind of figuring out what they're going to do. So please make your appointments with me. Let's get talking. I've talked to a lot of you, and some of you, I don't know who you are. So come and talk to me, please. All right. Thanks again for coming, you guys, and supporting our program.